Talk about yes, please come in. So today we will talk about the pathogen that cause the GIT infections. Okay, and these bacteria are mostly commonly and uncontaminated foods. And today we will see the fried rice, which microbe is responsible. As we eat here in the Middle East or in the Western countries, they eat most of the time fried rice, okay? And similarly, raw, milk or pasteurized milk. But sometimes due to technical defect in the machine, in the processing, at that time the food can get contaminated. You might have heard this in the Saudi a few years back. A number of cucumbers were thrown into the sea because of infection of the E. coli. E. coli was present inside the cucumber because at the time of pollination, the bacteria enters into the, at the time of fertilization, at the time the destroys. Similarly, just yesterday, I guess maybe you have heard this news, Listeria monocytogenes. There is a Nestle is a company, a milk product company, right? Nestle, you heard the name of Nestle?
Yeah.
It makes the ice cream. Yesterday they removed all the ice cream from all the stores in the America. Why? Because in few of the drumsticks, the cones of ice cream cones, drumsticks, they found the presence of the Listeria. So they removed because this is also, Listeria also causes the infection in the GIT. And about the Listeria, this microbe was found in 1920, okay? 1920. But first time this disease in the GIT was found in 1981. After how many years? Around after 60 years, we came to know that this bug, it causes the disease in the humans. Before that it was related to disease, but that time, later on in 1981, it was found in the pregnant ladies. When studies were done, this microbe can also cross the placenta barrier, okay? Listeria. And transmission of these microbes through food, okay? Food and water. Transmission of those microbes. So some of them are gram positive, some of them are gram negative. So let us see about, you already had a lecture on their pathogenesis, on the structure, other things. Today we will see about their, how to diagnose, how we can diagnose them. Salmonella typhi, the first microbe. It's a waterborne disease. Even if you see Bangladesh, India, Nepal, Pakistan, not well sanitation systems, you will find this microbe, you will find this bacteria. So please take care, the flame is on. Maybe by mistake it will fall down or something like this. So how to identify this salmonella? Collection of sample, it has I did not set this. Every time when we go for the sample collection, there are certain procedures, okay? I did not set it because already you have been taught from the first year that there should be the gloves, proper site of collection of the sample. For example, somebody is having a urine infection. You cannot write as a doctor, go and test for the blood, okay? Proper sample, proper collection, proper location is important. So when we have a salmonella, this microbe, to identify this microbe, it depends on the case of the infection. If it is a case 1, week 1, just recent infection, you can take blood as a sample, okay? If it is a week 2, because we get this information from the patient, okay? When we ask him how long you have been ill, or something like this. Week 2, it is in the serum. Week 3, in the fecal and in urine. So first, usually the blood is considered as a right time to diagnose the microbe salmonella. And if you go for the gram-negative gram scan, you will find it's the gram-negative and motile. It has a many flagella, peritricus, this microbe. It is catalase positive and oxidase negative. So what is the catalase positive? Quick.
Catalase.
Yes. What is the catalase positive? You are the third year.
Hydrogen bricks H2O2.
Correct. the hydrogen this microbe is catalase positive. This means this bacteria having an enzyme, the presence of catalase in this bacteria, it attacks on H2O2 hydrogen peroxide and makes it to water and oxygen. Arrow up, we use the gas bubbles. Okay. Now we will see. Please give me this one. This is hydrogen peroxide. I put it on a slide just a few moments before when I enter this hall. Okay. This is hydrogen peroxide. This is hydrogen peroxide. And now I put in a colony of a bacteria. This is catalase positive. Okay. Now we will confirm this bacteria has catalase present. Okay. Catalase enzyme. We will pick up a colony. Okay. This is the colony in my hand. And what I will do, some students are closer, they can see, I will put it here. You see bubbles coming out?
Yeah.
Please come and see. You see bubbles coming out? This means this bacteria has an enzyme catalase and it breaks down hydrogen peroxide into water and oxygen. These bubbles coming out, this is oxygen, okay? Fine?
This is catalase positive.
This means this microbe is catalase positive. Please do it fully to me. Clean. You know, once it is red hot, finish. There is no bacteria at all. It's over. So now you have seen, you see here bubbles are coming out because the colony of the bacteria is situated here. And this means when the bacteria makes interaction with the hydrogen peroxide. What is the job of hydrogen peroxide?
Breaks out the
Hydrogen peroxide destroys the cells. Okay? Even in our body, we have the free radicals, they damage our cells. Similarly, they try to damage it. But when bacteria is having the catalase enzyme, it releases it immediately and neutralizes the hydrogen peroxide into water and oxygen. The bubbles that are coming up, they are bubbles of oxygen. Please, if you don't want to switch off the gas, this job is not over because you cannot keep it risky in the lab. Fire issues. So this is microbe is catalase positive. You see the bubbles here? It's a positive and it's negative. Now what is oxidase? If this microbe is oxidase negative, what is oxidase? You forget, huh? Okay, you have a lot of things to do. Oxidase again is an enzyme which is present in the bacteria. The job of oxidase is to transfer electron. Similarly, in our mitochondria, we have cytochrome, okay? We have a cytochrome. When it transfers the electrons and we make respiration. If we are respiring, our mitochondria, if it produces the energy, you might have heard in the biochemistry, mitochondria is the powerhouse of the cell. Because why? It produces energy ATP. How? Electrons transfer from cytochrome. Cytochromes are the protein-like structure. Cytochrome A, B, C, D, E. 5 cytochromes in our mitochondria. The electron shifts 1 to 2, 3. Finally, at last, the electron goes to the oxygen. It reacts with oxygen and forms the water and the energy. In our case, similarly in the bacteria, if bacteria is having the oxidase positive, that time the bacteria has a capable to perform an aerobic respiration. Form an aerobic, because oxidase enzyme reacts with the oxygen and form the water, and you will find the microbe oxidase positive. Usually, all the Enterobacteriaceae family, okay, family are oxidase negative. They are anaerobes. Usually, they are oxidase negative. Okay. This means when microbe is oxidase positive, this means there is presence of enzyme. It reacts with the oxygen and form water. In this case, the microbe is oxidase negative.
So it helps in aerobic respiration of the bacteria.
I told you in aerobic case of respiration, bacteria are having the oxidase positive.
Ah.
Oxidase positive. Apart from this, God has given money. For example, we are aerobic, right? We are aerobic or anaerobic? We are aerobic. We are taking the oxygen and it produces energy in our body and carbon dioxide goes out, right?
Yeah.
We take oxygen as a fuel, and with the help of the mitochondrial cytochromes and it produces energy in the ATP that we are utilizing. Each cell in our human body has a mitochondria, it produces energy, so we can walk, we can talk, we can speak. So similarly, but for the microbes, they are either aerobic or anaerobic. Sometimes they are facultative. They may survive both in oxygen and in absence of the oxygen. Now, if you go for the culture for this microbe, Salmonella, you need better to go for first broth culture. Okay? These are the blood culture bottles here. This is called as a broth culture, liquid. Two types of media is there. Broth culture and solid media. Broth culture and broth culture media and solid media. Usually what we do, first we do this on the bottle culture, these culture bottle, we put the from the patient sample. For example, you withdraw the blood from the patient, okay? You bring it to the laboratory and you put the blood sample collected from the patient, you will put in these bottles, okay? And this is the incubator. And you put these bottles here for a night, 24 hours, okay? But in this case, Salmonella, sometimes it may take 2 days, sometimes it will take 3 days. And when you see this culture bottle after 2 or 3 days, if it is clear, same as it was the first day, so there is no infection. But if there is a turbidity, it is opaque, change in color in the media, okay? Then what you do, you put it on a culture plate, on a solid media to find the bacteria, it's whether it's the gram positive, gram negative, and other tests we usually do. So, and after the culture, if we do the Salmonella culturing on the McConkey, you have heard about the McConkey culture?
Yes.
What features about the McConkey? Please tell us.
Non-lactose fermenter.
Correct. Lactose and non-lactose fermenter. Because lactose, the McConkey agar, it has a property, it inhibits the growth of, it inhibits the growth of all gram positive bacteria. Okay? When you put the Salmonella on the McConkey agar, the bile present in the McConkey, it will inhibit the growth of gram positive. This bacteria is gram negative, okay? So, after 24 hours, the growth will be seen as non-lactose fermenter. As non-lactose, you see here, no color. If it is a lactose fermenter, the color will be
Pink.
pink. Why?
Fermentation.
Fermentation, correct. There will be the fermentation, there will be a decrease in the pH and you will see the bacteria is lactose fermenter or non-lactose fermenter. So for Salmonella on the McConkey, it is non-lactose fermenter. Okay? It is non-lactose fermenter.
So, doctor, you put it in the broth, and if there is turbidity, it means
Then
that
you
there
do
is
one
growth.
more color. There is growth. Turbidity means there is growth. Okay?
Then you check for McConkey agar.
Then we go for the McConkey. If it is a non-lactose ferment, confirm this is Salmonella. And another test I told you, catalyst already shown. This is one more culture media. If you want to more tests, okay? Once again, more confirmation. XLD is a xylose lysine deoxylate agar. What you do, again, you culture this sample. Even if you know, for example, as I told you, week-wise, I want to go back here, blood, serum, stool and urine, okay? So here in the XLD, if the doctor advises for the stool collection from the patient, you do it first on the 7-night broth in the liquid, there is opacity. Next day you will do culture on XLD, okay? On the XLD, the Salmonella produce black colonies. Why? Because in the XLD media, there is a thiosulfate, a chemical, okay? there is a thiosulfate community. That thiosulfate is utilized by the bacteria, okay? And bacteria produces the gas, hydrogen sulfide. It gives this black color, okay? You are not going to remember the mechanism, how it's a black color. Usually the salmonella on XLD, it produces the black color. If somebody asks why, the reason is the bacteria produce a hydrogen sulfide gas because due to the metabolism of thiosulfate present in this medium. The colonies are black. Now Vidal test, you may have heard about this. Vidal test is usually done on serum dilution, or you may have agglutination test. So Vidal test, when we say Vidal test, we are talking about the serum antibodies, antigen and antibody. To detect the antigen and antibody in the salmonella in this bacteria, we do two types of tests. One is a tube type, another is slide test. When doctor asks the patient go for the Vidal test, usually doctors advise Vidal test. Then it's the job of the laboratory whether he will do a tube test or slide test. Depends on the availability what is present in his lab. Okay, if he wants to do, because this is very quick test, and here he needs to do a little labor to perform the other test. Usually what we do in the antibodies, IgG, M and IgG. The bacteria here, salmonella, it has a flagella, okay? It has a flagella. Antigens are O for somatic, antigen of salmonella type. Somatic here, this is the O antigen and this is the H. Flagella is the H antigen. Two antigens are here, O antigen and H antigens.
VI antigen also.
So sorry?
VI antigen for the capsule.
O for from the body of the microbe and H for flagella. These antigens, they are used to confirm the presence of the bacteria. It is present or not in the blood because it reacts with the antibodies. Yeah, antibodies. And why only those two? Because the O and attribute IgGM antibody and G to antigen. Okay, this is the reason we are using these two antigens. You got my point?
Oh, attaches with what?
IgGM and IgG for H. So what we do to confirm this test? This is O antigen. O for the membrane, the body of the whole bacteria. H for flagella. H for? This is the bacteria. This is the somatic O, and this is the flagella H. Antigens. And here IgGM will bind your IgG because we cannot see it. We will put the antibodies and see. What is AH? No, for para A and para B. We have salmonella,
Salmonella paratyphi and typhi.
Correct. Salmonella typhi and paratyphi are other two strains of the same bacteria. Okay. Sometimes a patient is having salmonella typhi only strain, but in some patients you may have both salmonella as well as paratyphi, paratyphi A and paratyphi B. So this is the paratyphi A H. This is the paratyphi B.
Yeah.
Okay. This is negative control. This is positive control. I want to ask you a question. You had a lecture on this.
For the typhi.
Yes, okay. Masalan, this is the salmonella typhi, okay? This is O antigen, this is H antigen, correct? Now we have a paratype. It is same O antigen and H antigen, correct? Understand? So now tell me, why in salmonella typhi we do O and H, but why in paratyphi we do only H? Why we do not O? You got me question what I want to say?
Yes.
If salmonella, you had a lecture, salmonella typhi and salmonella paratyphi. Okay, both salmonella species or strains, whatever you would like to say, having O antigen and H antigen. O for somatic body, H for flagella. Now tell me, when we do the Vidal test, we do for O antigen and H antigen. For Salmonella typhi, O and H, Salmonella typhi. But for paratyphi, we do only H and paratype A, we do H, paratype B, we do H. Why not O? Why not paratype O, paratype B? Yes, paratype B, O. You got my point what I want to say?
Maybe it doesn't affect the result.
Because it interacts this O or the paratype B, it will interact with this O. It may give you the wrong result. Because of the similarity. Cross reaction will happen. Paratype is O is similar to that of Salmonella type. So this is the reason in the paratype we exclude this O antigen. We only keep H. So it is shown here H. Paratype AH, paratype BH. Salmonella type O, Salmonella type A. Here now you see already the antigens are here. These antibodies are here on the plate, and when you put the sample, serum sample, it depends on the clumping agglutination. If agglutination happens, you will say it is positive. It is positive. The presence of here in this slide, it has been shown this patient has infection of Salmonella typhi, O present, H present. In the para, it is also present. Okay, but in most of the patients, you will not find the para strain present, but sometimes it is present. Okay, but main what the infection, the main infection in the usually in the typhoid is because of the Salmonella typhi. Salmonella typhi. Now comes to the serial dilution. What is the serial dilution? I will tell you about this and remember for all. One is for all the serial dilution. Count these gift proteins, this will confuse you. I will start from here, okay? 1, 2, 3, 4, 5, 6. Okay? Do one thing, you will take the normal saline, okay? Normal saline is how much? Correct. Normal saline is isotonic solution 0.9. If you give a human body normal isotonic solution, it will cause no problem. If you give hypotonic, you know the results. So we will take isotonic normal saline here. Here in tube number 1, 1.9 ml. How much? How much? In tube number 1, in tube number 1, we put normal saline 1.9 ml, okay? From tube number 2 to tube number 6, from 2 to 6, we put 1 ml. How much? I hope you are understanding it, okay? From here 1.9, okay, you put the normal saline, 0.9% of sodium chloride. To perform this test, okay? To perform the Vidal, this is finished now. Yes. We'll talk about the Vidal test now. So next step, what you will do, you will put serum of the patient, okay? Of the sample, you will put the sample here, plus 0.1 ml. How much ml total? 2. Correct. This is now this tube 1. This is the sample. I will write here sample. Okay? 0.1. This is now 2 ml. Now let us see what is a serial dilution. From this tube, you will put 1 ml and put it here. Here you will do nothing. It is just 1 ml. But from here, you will put 1 ml, give it to him. How much it become now? It will become 2 ml plus 1 ml from this. But again, you will remove from here 1 ml due to here. It will remain again 1, it become 2. Again, you will give it to here. It became 2 ml, but when you remain, it became 2 ml. Keep this last one, number 6 aside. Do not do any, do not make this here. Now you have 2 ml here, right? Take 1 ml, throw it out. Okay? Throw it out. This means you are making the serial dilution of serum dilute. We diluted the serum because here is 1 ml, 1 ml, 1 ml, 1 ml. The sample was total 2 ml. From here we collect. When we add this concentration, it becomes double. From here, when we take and it becomes the double of this. What was here, it becomes the double of this. This means the concentration is increasing. So here you see. Here there is no sample tube. Okay, first tube is not there. It starts from the 2. So first is the double 20. It will become 40. 40 become 80. 80 become 160. 320 and doubling. So on, so on. So then what you do now? In the point, these 2 steps are clear. And point number 3, you put few drops of, or 1 or 2 drops of antigen. Antigen. You have 2 antigens, antigen O and antigen H. So what you have to do for this serial dilution? If this is for O antigen, okay? The same serial dilution you will make. The same. Similar method for H. Okay, second row of tubes. First row of tubes for O, and you make third row for correct AH. Fourth row for here. Same procedure, nothing to do. But in the third step you will do. Here you will put O antigen. Here you will put H and not antigen, anti-serum. Here you will put antibody. You will put antibody against H. You will put antibody against A, antibody against BH. Then, when you have added, then you start seeing. What you start seeing? The clumping in here. So at this concentration you have, okay, here are antibodies present, but not much. 20, 40, 80. And in 60, it gives you the highest value. It gives you that. And in the lab test we found such a condition, we wrote the patient is having an active infection at this 4-fold. Okay, fine. But if it is lower, we can say weak infection. If it is at 20, no infection. Okay. If you go up again, you see here, OH greater than 160, active infection. For flagella, it may be IgG. It is 160. It may be the past infection. Okay. So this is about the serial dilution. You understand the serial dilution now? Okay? Fine. So what is the principle of Vidal test? Principle of Vidal test is the agglutination of antigen and antibody reactions. Okay? IgGM and IgGO, they interact with flagella O and H.
The antigens are commercial, Doctor. You put it.
Sorry?
The antigens that you put are commercial.
Of course. Antibody.
The antibody or the antigens?
Antibody. We get the patient sample.
Yeah.
When we take, usually you see here.
And then you put the antigen.
This is the sample.
Yeah.
Okay? This is the sample. But step number 3 you are asking, right?
Yeah, when you put the antigen.
When we put the drops of the antigen again of O and
which is commercial antigen.
Commercial. They are available. There are kits, Vidal kit. If you go and see in the laboratory, Vidal kit, it is available there. Okay?
Whereas the antibodies are preformed in the blood.
Already prepared. They were already
in the blood.
Made. Yes, they are already made from the laboratories. There are many laboratories. One of the best laboratories is Tulip. He made the antigens. And Tulip. Maybe you have heard this name. Okay? It's an American company. Makes these antibodies and antigens and sell it to the market. Okay? So salmonella is over. Any questions regarding the salmonella? So we have XLD for stool. We have non-lactose fermenter collies. It is an oxidase. It is a catalase positive, and Vidal test, tube test as well as the slide test. Bacteroids. You heard this name before? Actually, this microorganism. This is actually. He's a good guy. He is not a bad man. Who? This is present in our gut already. Normal flora commensal. And good thing scientists say the good thing about the bacterioids. it will not allow to grow the harmful pathogenic insect because it has a so good activity fast inside the human body. Even it has been said it does not allow salmonella to attack very easily until and unless it becomes weak. So this is a property about this. But sometimes if it is out from the gut, if you go internal abdomen in any part of your body, immediately it will cause the infection. It will cause the disease. So, for example, if somebody have some problem inside, ulceration or something, if this bacteria makes entry, it causes the infection. God forbid somebody is having the accident or something like this. If the gut is injured by accident or something, this microbe makes the entry and causes the infection. Bacterialis fragilis. Even if you see them in the market, we get the sachet of probiotics. This bacteria is present in this. If you read the sachet on the pharmacy store, as a source of you have the probiotics. You heard the name of probiotics?
Like in yogurt and
Correct. Like in yogurt and other things. So actually it's a good, but it become bad. If something bad happened to the human, then he became the enemy that time.
so it causes opportunistic infections. Yeah.
Yes, you can call it opportunistic infections. When he get a chance, okay, it causes the infection. But it should move from the gut. But usually it's present there from the evolution of the bacteria. Okay, you see, you have to collect the abscess. If it is abscess or if it is any kind of energy injury or something, you can collect the sample and send it to the lab. It's a gram-negative bacillus. It is a non-motile, obligate anaerobe.
What's
Gram-negative bacillus.
obligate anaerobe?
What's the obligate anaerobe?
It's like
Obligate anaerobe, it cannot survive in presence of oxygen. Finish. It will die immediately. Okay? Obligate anaerobe, it should be present in absence of the oxygen. So if you need to grow this bacteria in the laboratory, you need anaerobic jar. Okay? That will stop oxygen supply to this microbe. This is actually black color, huh? And if you grow the bile-asculin agar, you will find complete, these are the black colonies. You will find the black colonies. And gentamicin is added. Why? It inhibits the growth of facultative anaerobe. What is the facultative anaerobe? Facultative anaerobe is a anaerobic bacteria or those bacteria that do not survive in presence of oxygen. Anaerobes, okay? They do not need oxygen for their growth. They need carbon dioxide. Facultative anaerobe means the bacteria has a capability to grow in both oxygen and in absence of oxygen. Bacteroides fragilis, this bacteria is an anaerobe. This bacteria is an anaerobe. It does not need oxygen. So we add gentamicin and bile so that it will inhibit the growth of other bacteria, other facultative anaerobes. Those bacteria who have a capability to grow in absence of oxygen. To stop the growth, we add the gentamicin and bile so that only the growth of Bacteroides fragilis is enhanced. Okay? Listeria. Many things to collect. As I told you, in 1960 this microbe was first time found. But its diseases to humans as source of the food, even through milk. We have a milk here in the market. It can be passed through this Listeria. It was found in 1981, first time that this bacteria caused GIT infections. It's a motile, it's a tumbling. What is a tumble?
Like states?
Tumbling is
Like states.
yes, tumbling is like this. Like this, okay? This microbe, it shows the movement tumbling because it is of a V shape. It's like this. Usually bacteria rod shaped, okay? But this bacteria you see here, like here it is a comma shaped Listeria. Here it is like this bacteria, comma shaped. So due to this V shape, when it moves, it makes tumbling. it tumbles, okay? This bacteria is catalase, positive, the same thing, and oxidation negative. The sample you can collect from blood, from food, as I told you, listeria, it can pass the placenta also. Okay. The culture for this bacteria, for this microbe, small zone of hemolysis you can see. To confirm this listeria LM, listeria monocytogenes, you see the Muller-Hinton. This is the Muller-Hinton agar, okay? This is Muller-Hinton, but when you add blood to it, when you add sheep blood, Muller-Hinton will, when you add the sheep blood, it becomes like this. This is the Muller-Hinton, and when in the laboratory, when we add the blood to it, sheep blood, it becomes like this. Okay? Now on this culture media, if you put the listeria, and keep it in incubation for a 24 hours, next morning you see the growth of bacteria, okay? And when you remove the colony using this loop, you will find the clear zones. Here. Small zone of hemolysis. Behind the colony. You have to remove the colony, then you can see the hemolysis. Not complete, okay? Not complete hemolysis, but hemolysis is there for listeria. And one more, this is called the Anton test. You have to catch the rabbit or guinea pig, okay? Usually the rabbit, if you put this in his conjunctiva, this listeria, within 24 hours, there is conjunctivitis. Within 24 hours. So one more proof for this listeria, okay? One is this lab test, Muller-Hinton sheep blood agar, one. Second, this is Anton test, and first one is simple. These are called the biochemical tests, catalase, oxidase.
What about the last line, motility and hemolysis differentiated?
Is the motility and hemolysin differentiated from coronary bacterium. I just put it in brackets just for information, because coronary bacterium is not our topic, okay? Just for information. Helicobacter pylori. You heard this name, Helicobacter pylori, okay? This microbe is a gram-negative bacillus. It usually lives where it causes infection.
Habitat for stomach.
It is present in your stomach. Now tell me, what is the pH of a stomach?
1 to
0.5 to 1.5.
Okay, we can say 2, okay? pH of stomach is, even if you find in the exam, like 1.1, 1.8, if 2 is there, immediately keep 2, okay? Because pH of stomach is 2. This means highly acidic. Highly acidic. But the question is, why this bacteria survive in the stomach?
Because it forms a base. It forms an alkaline.
From where? Because?
From ammonia. Urease enzyme.
Okay. Because this bacteria has a urease. When this scientist, he was a doctor, when he said in the stomach there is a bacteria, Helicobacter pylori, it was previously called Campylobacter. All the faculty in the university laughed at him. When he said the Helicobacter causes infection in the stomach, all the faculty laughed at him. You know why? They said, how come? pH is a 2. No bacteria survives. How this microbe can survive? Then you know what he did? He took the this Campylobacter and take it inside and started test on his own body. And when he proved it, he got the Nobel Prize. You know, when he proved it, he got the Nobel Prize. He proved that this microbe has presence of the urease enzyme, and now we will see in the next slide what urease does. So usually to find this microbe, you need a sample, either stool, blood, or gastric biopsy. But the best sample is a gastric biopsy. Okay, sample from gastric secretion. You have to put the tube inside and make a punch on your stomach inside and collect some piece of tissue from the stomach, okay, to confirm this microorganism. Then you can do one test, send it to the laboratory, in the microbiology laboratory. There are some kits available, change in color will indicate infection is present or not. So this microbe is gram-negative bacillus, it is motile, microaerophilic. What is microaerophilic? Remember these small terms.
It needs a trace amount of oxygen to survive.
Traces amount, little amount of oxygen.
But if there's not if oxygen is not present, it will die.
No, it will not.
Independent, right?
This means higher concentration of oxygen, it will die. Higher concentration of oxygen. You see this catalase? Positive. This means catalase enzyme is present. Oxidase? Positive. And urease? These are the three properties of this. I said Enterobacteriaceae family. This is not from Enterobacteriaceae. Okay? Do not remember Enterobacteriaceae only gram-negative. There are many others. Huh? Okay? Fine?
So it's not in the group, in the family?
Take a textbook and see the Enterobacteriaceae family separate. E. coli, Salmonella, other. Okay? This is a gram-negative family, but away from the Enterobacteriaceae family. Okay? Because there are some belong to firmicutes, some belong to spirochetes, some belongs to others. Okay? Fine?
Okay.
This should be very clear. Enterobacteriaceae family or separate list of microorganisms. This is also gram-negative, but different. So this microorganism is a catalase positive, oxidase positive, urease positive. Three properties, strong properties in this bacteria. Now you see what it does. What this microbe does inside the body. This microbe has an enzyme. Which enzyme?
Urease.
Urease. Okay? This is a urea. This is a urea. When urea is in your stomach, through food, okay? And when urea component is present in your stomach, and this bacteria is also present, it will release the enzyme urease. Okay? What urease will do? Urease will break this. Okay? Urease will break this. It will come, carbon dioxide will come out. Carbon dioxide. And plus ammonia. NH2. Two molecules of ammonia will come out. This ammonia is a base. This is a. What is the pH of stomach? Correct. When this microbe breaks down the urea into carbon dioxide ammonia, and ammonia is a base. Ammonia is a. Here is pH 2. When the base reacts, when the base comes to here, it increases its pH more than 3, 4, 5. It increases the pH, okay? Or it neutralizes the pH. It can neutralize the effect of this and it will help to grow the bacteria. Bacteria will grow because of this base. It broke upon this. So this on that basis, on this mechanism, the doctors, scientists, academicians, they made a test in the market available kit. It is called urea breath test. Okay? C for carbon. C for? Why they choose carbon? Because carbon dioxide is here. Okay? I told you carbon dioxide would throw it out. And the kit available, it is C13. C13 is a radioactive carbon. Okay? I will tell you the principle, what they do. See at this table. This is human, okay, person, and he is taking the urea, C13. He took the urea inside. The bacteria is present. This bacteria is an enzyme urease. It attacks on the CO here, and I show you CO, carbon dioxide. It is broken down, the carbon dioxide ammonia. Ammonia neutralizes the acid in the stomach. The carbon dioxide goes out to the lungs and come out. And here is a Euro bag. Sorry, not Euro bag. It is called the carbon 13 breath bag. We ask the patient, do like this. The patient throws the air into the bag, and there is a readings, change in color in the bag. It will change. This means the microbe is present. So what we do in this test, we give the patient urea, but radioactive. Okay. radioactive. But this radioactive will not affect the human body, because radioactive elements affect the human body, but this it will not. It is not harmful. We ask the patient to drink urea with C13, and the patient drinks it. Okay, if bacteria is present, you see it can detect the active infection. The patient drinks the urea solution labeled with isotope of carbon-13. H. pylori, the bacteria converts urea into ammonia and carbon dioxide. This carbon dioxide will throw into the bag and it changes the color. This means microbe is present. And this urea breath test kit is also available in the market. Okay, it's available in the market. Again, this is what's the principle? The principle of urea breath test is very simple. The principle is the urease enzyme breaks down the urea into carbon dioxide and ammonia. Finish in a one line. Urea breath test, the principle is breakdown of urea into carbon dioxide and ammonia. Carbon dioxide goes out through respiration. Ammonia is in the stomach. It neutralizes acidic nature in the stomach, or will increase the pH of the stomach. That will help to microorganism grow. This is the reason it is very dangerous microbe, pylori. And from where you got this infection, you know from where? From eating more burgers and other
Spicy food.
not spicy food and other the barbecues. The favorite site is burger. Favorite site. KFC, Almake, no sorry, not Almake.
Not Almake,
McDonald's. Okay, so be careful. Do not focus more on the burgers. Okay, because you don't know when the bacteria will come. I was telling you listeria. Just yesterday in the newspaper in America, from all the stores they remove ice cream. Why? Because they found the listeria was present while processing. When they were packing the ice cream to get market, and they found it. But I'm sure if this case happened in India or Pakistan, nobody cares. Let people go. Nobody cares. Okay, this is the culture for it. If now you want to culture this microbe, Helicobacter, or you can say Campylobacter, you need the incubation for 3 to 5 days, and you see the very light, thin colony, light color, transparent colony there. And the selective medium from gastric specimen, you need to sample from gastric lavage or whatever you take it, you put it there and you find the results after this. But usually I put this test before because more important tests come.
This culture is called modified medium.
It is a Helicobacter agar modified medium. This media is prepared
for
as per the needs of
specific for the Helicobacter.
Fine. Any
Yes.
question? Sure. Staph aureus. This is the bad boy in the microbiology. Very bad boy.
Everywhere you find.
Everywhere you found this guy, and very notorious guy. He has number of exotoxins and cause so many diseases. And the most important about this bug, no, forget about food poisoning. This is now antibiotic resistant, MRSA, methicillin resistant Staph aureus. In the hospital, when we doctors do the surgeries, after post surgeries, number of infections, number of deaths are caused by this microbe. Some patient is operated, okay? He will not die from that disease which he was having, but he will die from the infection from this MRSA. Here again Staphylococcus aureus, but here today our topic is caused the food poisoning. It caused the food poisoning. It released the toxin, exotoxins into the food. When you keep a food for a larger period of time, exotoxins are released and you get infected. One. Another, if you get the sufficient number of bacteria, more number of it is not necessary that bacteria go inside it cause the food poisoning. Even the toxin produced by the bacteria. The toxins are two types. The toxins are. This is last year I have written it here. It is similar like this. Nobody uses board except me. You see, this is, for example, this is the exotoxin, okay? Sorry, this is the endotoxin. Endotoxins are usually endotoxins from endotoxin from the membrane, and they are usually all present in gram-negative. Okay, finish. Exotoxin comes from the bacteria inside and come out. Exotoxins come from the bacteria. So when somebody is having the food poisoning because of Staph aureus, there may be two reasons. Either the Staph aureus go inside his body, one, or the Staph aureus release the toxins. Because why? Everybody knew we eat food and we go to the washroom. Okay? What is our metabolic waste? Is the toxin that we remove. Either it is a stool or a urine. Fine? Similarly, when the microbe eats the food, the things that he needs from the food, at the same time he produces a toxin. We call them exotoxins. We call them, and these exotoxins are short proteins, polypeptides, few amino acids, and they come in the form of vesicles and come out and they spread in the food, and we don't know. And when we eat it, we start vomiting, diarrhea. Sometimes it may go beyond the dysentery. The more bad it is, and we need to go to the hospital that time. So Staph aureus is one of them. Not only this, I told you it's many other diseases, but it's also called the food poisoning. You see, grape-like cluster, you know it very well. It is Staph aureus in the form of clusters. Just like the grapes, a bunch of grapes. Catalase positive, okay, and catalase positive, coagulase positive. Catalase positive, and the main difference between Staphylococcus and Streptococcus, what?
Coagulase. No, catalase.
The main difference between Streptococci and Staphylococci is catalase. All Streptococci, catalase negative. Staph, catalase positive.
Yes.
Remember this for lifetime. But all Staphylococci are coagulase negative or coagulase all Staphylococci are coagulase negative except Staph aureus. Remember this whole vocabulary. Staph aureus, A-R-E-U-S, it is positive. You have saprophyticus, you have epidermidis, all Staphylococci are coagulase negative. Only Staph aureus is coagulase positive. What is a coagulase positive? Coagulase is an enzyme produced by the Staphylococcus. When it causes the infection inside the body, it changes fibrinogen into fibrin. When it is present in your body, it releases the coagulase. And what it does, it inside around his surrounding, he coagulates the area. Fibrinogen gets converted into fibrin. Why? To protect itself. For example, I am in the center and you 5-6 boys may be around, so I am safe, right? Something like this. So this is catalase positive, coagulase positive, Staphylococcus aureus. And blood agar, if you culture the Staphylococcus aureus, if you grow the Staph aureus on the blood agar, you will find hemolytic, beta hemolytic, big colony is yellow. Now tell me why it is golden yellow color here. Oh no, I will ask you later, not now. Okay, this is beta hemolysis, yellow color on the blood agar. On the when Staph aureus, if you grow Staph aureus here, on the blood agar, grow on a blood agar, next morning you will see beta hemolysis, yellow golden colonies. If you, please excuse me, and it is about blood agar. If you grow Staph aureus or mannitol salt agar, the colonies will be yellow. It produces yellow colonies. Now tell me, what is the difference between yellow in the blood and yellow in the mannitol?
Reduce the pigment.
Where?
That.
In the blood or the mannitol? In the blood, it produces the yellow colonies.
Okay.
On the mannitol, it produces the yellow colonies. What is the difference between here? Yellow, it is the same or different?
It's not in this.
They say destruction. Why there are yellow colonies? Because of pigment. In the blood, whenever Staph aureus is grown on the blood agar, when it is grown on the blood agar, it produces beta hemolytic colonies as well as yellow coloration. At that time, microbe produces the pigment. This bacteria produces pigment. But when you grow on mannitol salt agar, the colonies are similarly yellow. How? You got the answer? In mannitol salt agar, when you grow the Staph aureus, the actual pH of MSA, mannitol salt agar, is 7. Okay? But when you grow the Staph aureus, it causes the fermentation of the mannitol and decreases the pH. Decreasing pH causes the change in color. These are the two differences for the Staph aureus. On blood agar, colonies are yellow due to pigmentation. On mannitol salt agar, the colonies are yellow due to fermentation of mannitol. pH is decreased. And one property of mannitol. And second, the mannitol, why it is called mannitol salt? It contains 7.5% salt. Usually most of the microbes die at the 7% of salt concentration. Okay? At 7%, so Staph aureus grows in 7% of salt concentration. Usually microbes do not survive under high salt concentration. This is the reason mannitol salt agar is used for growth of Staph aureus. Any questions? Staph aureus done?
Yes.
Catalase positive, coagulase positive. On the blood agar, beta hemolysis, golden yellow colonies due to pigmentation. On mannitol salt agar, it is yellow due to fermentation of mannitol. Mannitol is a sugar.
Yes.
Bacteria uses the sugar as a source of energy and it produces the acid in the medium and pH is decreased. When pH is decreased, color changes. Why color changes? Because this media contains a pH indicator dye. It contains dye. When pH goes decrease, it automatically changes the color of the dye which is present inside the media. Bacillus cereus.
Last one.
Whenever you go to the restaurant, please, usually ask him. If it is not a reputed good one. Bacillus cereus, it is a property when you have fried rice, okay, or fried food. Usually it has been seen. If it has been kept for 6 hours or 7 hours as such, after preparation, there will be the growth of Bacillus cereus. It will get doubled. Okay? It will get if Bacillus cereus is present there. And if you keep the same food for one night, okay, for 24 hours, he will have around 1 billion bacteria in a one night. About this. So Bacillus cereus is usually it is present in the fried food. If it is present, yes. This means contamination. Usually they keep the food safe. Otherwise, if it is present everywhere, so nobody will eat the fried rice. So you need a sample food. It's a gram positive, non-capsulated. It is aerobic bacteria. It is motile. Catalase positive, oxidase negative. On the culture, you will see the thick, grey opaque colonies of bacillus. Fine? As I told you, I found the same strain but different, non-pathogenic. But when I grow this in the laboratory here, it was the same like this. No difference. All properties same. What the properties are here, but only difference in their toxins. Here the toxin of Bacillus cereus called food poisoning. The other it does not. Fine, Bacillus? Clostridium. Again, one more pathogen that causes the food infection. It is catalase positive, oxidase negative, and spore form encapsulated non-motile. Any other property here? The identification of Clostridium perfringens. because we have Clostridium perfringens, Clostridium botulinum, right? Clostridium
difficile.
difficile, yes, and we have sporogenes, Clostridium sporogenes, right?
Same as difficile.
No, they're different. So first we will go for the culture for Clostridium. The Clostridium, there are different species, but Clostridium, perfringens, when it is grown on the meat, robots and cooked meat agar in the culture media, the colonies become pink color. You see the pink color? Pink color of the meat. Here it is a pink color. But you cannot see, eh? Is it seen pink color? Slight pinkish. If it is a pinkish coloration, this means it is Clostridium perfringens. Okay? Clostridium perfringens. That means this microbe in the robots and cooked meat, this microbe degrades only sugars. Degrade? Only sugar, which is present in the media and gives its pink color. It's called saccharolytic. You see, saccharolytic. If it is not Clostridium perfringens, the meat will turn black. It will turn black. That means bacteria is digesting the proteins. They are called proteolytic. So to differentiate the Clostridium species, we use robots and cooked meat. For Clostridium perfringens, we use the robots and cooked meat. If it is pink in color, microbe is present, saccharolytic. And on the blood agar, it shows the hemolysis. 1 and 2 type of hemolysis. Complete hemolysis due to theta toxin and incomplete. Wider zone of incomplete hemolysis due to alpha toxin. So this microbe has 2 type of toxins also. It causes the hemolysis also. So Clostridium perfringens on the robots and cooked meat, it gives pink corneas. Why? Because it is saccharolytic. It is saccharolytic. Other species of Clostridium, they give color black because of proteolytic activities. It is saccharolytic. Saccharolytic means it breaks down the only sugars. It is not showing the proteolytic activity.
Onion sugar.
Only sugar. Saccharolytic.
Sugar.
Okay? Because what's a meat? Meat is a protein. Okay? It's a protein. Meat turns pink without digestion. You see here? Meat turns pink without. This means this bacteria is not breaking down the protein in the meat. If it breaks down, then it is Clostridium, but other species, not perfringens. Fine? It is the hemolysis because of theta toxin and alpha toxin. This is one more test for Clostridium perfringens. It is called a Naegele's reaction. Okay? This is called a Naegele's reaction. What is the purpose of this test? To detect the lethacinase activity. Lethacinase is an alpha toxin. Okay? Produced by Clostridium perfringens. So we can perform this test. Usually in the Naegele's reaction we have a media. Okay? This is the media. This media contains egg yolk. It contains, you see egg yolk yellow. Egg yolk, it contains the, what is the lecithin? You have heard this name before? Lecithin. You heard this name in biochemistry? No? Okay. Lecithin is a part of a membrane, cell membranes. Lecithin is a component of cell membrane. So in this media we put the egg yolk. This egg yolk contains lecithin, okay? Lecithin. And when we allow Clostridium perfringens to grow here in this culture media, the bacteria produces enzyme lethacinase. Okay? And it attacks on the lethacinase or alpha toxin by Clostridium perfringens. It attacks the lecithin present in the media and makes this type of form. Opaque. This means it has broken down the lecithin. Lecithin is a component of a lecithin is a component of a cell wall. If it is broken down
by
with
lecithinase.
the help of lecithinase produced by Clostridium perfringens is an alpha toxin. Okay, if bacteria produces this toxin, if it is present, it will break down lecithin and its structure will be opaque like groove.
Opaque?
Here you see here groove.
Yeah.
This is Clostridium perfringens here, this is here, but here it has broken down the lecithin. This is Clostridium origins, another no effect. But why here no growth? Why is it growth here? Here is no growth. No. See at the big plate. Because we have divided the culture plate into two parts. This is we divide this plate in two parts. Okay. This contains the media containing egg yolk. Lecithin is present in all here. Lecithin is present. We make the growth of bacteria here in this media. From this side the structure is like this. Lecithin is broken down, but in this side there is no it is not broken down. Why? Because we have added the antitoxin.
To the left?
To the left we have added the antitoxin against this lecithinase enzyme or alpha toxin activity. So this means when there is an antitoxin in this side, when bacteria produce the lecithinase, it encounters it with and stop it affecting the lecithin. Okay, but in this case, on this side, in this side, there is no antitoxin present, but lecithin is there. But enzyme of the bacteria, this lecithinase attacks it and it's like this. In this origin, there is absence of this enzyme. So you see it is growth here and here same, because when there is no absence, no presence of the enzyme, there is no breakdown of lecithin here. But here it is broken down because of lecithinase alpha toxin. In this side, it's not broken. Lecithin is not degraded. Why? Because of the presence of antitoxin. Clear? It was the name of the person was Nashka. He made this test to confirm. By this test confirms here that this microbe has capability to produce lecithinase alpha toxin, and this opaque, this structure, this means enzyme has degraded the lecithin. These are two similar names but different. Lecithin is a component of membranes, and this can be broken down by lecithinase. It is also called as alpha toxin in Clostridium perfringens. When this bacteria releases this enzyme, it broke down. But if we, on the other side, on this side, if we make the antitoxin here, it will block the, when bacteria produce the lecithinase, antitoxin will block it, right? It will block it and there will be just simple growth and there will be the growth, but there will be no activity of lecithinase, identify of this toxin. Fine? Understood? It is the time to go home and relax.